I purposely waited to start that recording. Okay, any questions? Okay. I haven't gone over with you chapter 9 or chapter 2, and so I want to do that now. If you haven't already outlined it, don't worry if you haven't outlined it, you're not alone. I've only got one set of outlines in so far. Okay. My chapter 9 is on joints and starts on page 209. It's chapter 9. If you don't have a book yet, I don't know how you're going to do these outlines. There's several ways to be ingenious about it, though. If you don't have a book in front of you right now, you might want to make notes. Okay, classification of joints. That's a definite yes. Fibrous joints, definite yes. Of course, you know these terms from our lecture a couple of weeks ago. Sutures, yes. Next page, syndesmoses, yes. Gomphoses, yes. Hopefully you're familiar with these terms enough that they won't take a lot of your study time. Table 9.1, absolutely, absolutely. It's very important table. Cartilaginous joints, yes. And the synchondroses, yes. I have not eliminated anything yet. Symphyses, yes. Synovial joints, yes. Figure 9.2, that's up to you. You don't need to outline it. It bears looking at, but you don't need to do anything else with it. Okay. Under synovial joints, we have general structure of those synovial joints. Yes, please. Flip the page. Figure 9.3 is a yes. Figure 9.4, dissection of the knee, is a no. I'm going to go over that with you on Thursday. Nerves and vessels is no. Articular disc is no. Bursae and tendon sheaths is yes. And although the rest of the chapter is interesting and not terribly difficult, this is where you're going to stop for chapter 9, for the outline. So if you find movements allowed by synovial joints, draw a line above it and write the word stop. Chapter 2 is the longest chapter we will have by this date. And I don't think I ever eliminate anything from the chapter. I'm just going to take a quick look myself. I'm on chapter 2, though, if you want to follow. And for my book, it's on page 22. The figure 2.1 is important. It's an idealized cell. No cell actually looks like that. A muscle cell certainly doesn't look like that. A nerve cell does not look like that, but it's a generalized cell. Okay. As you go through the reading, I'm on the next page, line 24, the plasma membrane. Yes, I want you to read that. But there's a table, 2.1, that lists all these same things again And you do not need to outline table 2.1, no. Okay. Because you'll get that information in the text. The plasma membrane, yes. Structure, yes. Function, membrane transport, which we talked about the other day. Cytoplasm. I'm flipping pages pretty rapidly here. Golgi apparatus, lysosome, all those are yeses. Peroxisome is a no. It's the only one I ever take out, I think. Peroxisome, when you get to it. Mitochondrion before that was a yes. Cytoskeleton is a yes. Keep going now. The nucleus. Okay, here's the part I don't want. When you come to the part chromatin and chromosomes, I'll be going over that today. I don't want you to outline it. And when you get to figure 214, the structure of DNA, we're not teaching genetics here. Oh, my genetics class was absolutely horrible when I was at UCI, taught by Dan Wolf. I still remember that guy. He was scatterbrained. You do not want a scatterbrained person teaching genetics. It just doesn't work out. I don't think any of us got it until we did our own reading. But we're not doing genetics here. That's not gross anatomy or anything close to it. Okay. So figure 214, 215, no. And when you get to the cell life cycle, draw a line above that, that's a stop. Okay. So I did eliminate some of it for your outline. It's always best to take the exams on the day that they're given. The only reasonable reason not to is if you're sick or you have an emergency that you have to take care of, and you'll email me about that so that I know to expect that you'll be taking a makeup test. Makeup tests are extremely hard on me, okay? And they're not any easier really on you because they have to be different than the test that was actually given. So I have to make up another test. That's the part that I don't like, of course. And you don't like the fact that you have to ask me for it. But if you have to ask me for it, ask me for it. Okay? Because fair is fair. If you're sick, fine. Not fine, but let me know. If you have our nemesis right now, COVID or anything like that, I think you should, if you don't think this through yourself, let the Office of Instruction know, let possibly the Chancellor know, or the Health Department at school know. I know if you're sick with that disease, you probably don't feel like doing anything. Maybe somebody else could let them know for you. It is going to probably have a bearing. I don't know how many people in our class have it, had it, may get it, or in any other class. But I think the sum total is going to decide, help them decide when really to open up again. Now it's still the 24th for us. It's still the 24th. It's a small, the classroom, it's really actually a good sized classroom, but not for 28, or in our case, 31 people. We're going to be close together. I certainly don't want to get it. I've got a lot of underlying, what do they call them? Help me out. I'm trying to get somebody to say something. Underlying factors. Okay. Well,
Conditions?
that's what?
Underlying
Said
symptoms?
symptoms, underlying symptoms. Do you think heart disease is one? I had a heart attack 28 years ago. I've been taking care of myself ever since. And they can't find anything wrong anymore, but they keep looking all the time. If we are able to go face to face, which is, really the champion way to teach, we're going to have to really manage, still try to keep apart from each other. It's hard to do. It's really hard to do. And if anybody does come in and get sick, I don't know what they'll do. They'll probably close it back down again. Anyway, we need to know. I think we need to know, if you're sick, tell somebody. Don't just tell your instructor. Uh, Go, like I said, Office of Instruction, Health Department. They're in the website. You can't miss them. Okay, and possibly also the Chancellor's office. They're the ones that are always sending out these messages. Okay. 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 Any questions or questions about that?
I just have a question about the outline. For chapter 2, what was the things you said not to outline before chromatin and chromosomes? It was like the first few things you said not to outline. Sorry, I started writing it down and
Did somebody else have that? Because I've closed my book up now.
You said peroxisomos or
Peroxisomos.
Yeah, those, and then figure 2.1 not to do.
Okay, got it.
Okay, thank you.
Okay, thank you. Because like I said, I closed the book up. And okay. Okay, then, if you don't have any questions right now, before we share the screen, let me remind you, please, get the practice quiz done. There's no reason not to. It doesn't hurt you, it only helps you. And share the screen. Okay. I'm a little shy of using this because of the one day we got stalled. But let's just keep our fingers crossed. We got it. There will not be a discussion section today, but I will have a discussion opportunity, let's call it, for review of the bones on Thursday. And anything else you may want to talk about too. Okay, so here we are down to week 3. Looks short. By the end of the week, it'll be quite long. The assignment is always the chapter outline to read. It's chapter 2, the one we just went over. Okay. that's the last of the chapters that you need to have in by next Thursday. Like I said, you can turn them in one at a time, two at a time, 3 in 1, all 4 at the same time. Okay. This is the last of what I call the remedial stuff that we have to do. Then we can get into real anatomy. Cell division. Okay. Now you're thinking back to high school, maybe even back as far as if you had a junior high school or middle school, when this was first given to you. Okay. Well, by now you've forgotten it. I have to reread it myself all the time. Okay. Cell division, it's divided into 2 different types, mitosis and meiosis. Okay. Mitosis and meiosis. Mitosis is sometimes called somatic cell division. Somatic means of the body, whereas meiosis is sometimes referred to as reduction cell division because the number of chromosomes is cut in half to form the gametes. The gametes each having half a complement of chromosomes. And when the gametes from the male and the female come together, it reconstitutes an entire set of chromosomes. The consequences or the outcomes of mitosis is that one diploid cell, now diploid cell is one that has 23 chromosomes from your mother and 23 chromosomes from your father. Diploid. It's abbreviated 2N. Mitosis leads to one diploid cell forming 2 diploid cells. There's no genetic recombination. A mutation could always occur, but we'll put that aside. Since there's no genetic recombination, the 2 cells formed by mitosis will be identical genetically. Okay. The duplication of the genetic material needed to double up and then cut in half to get back to normal. If that makes sense? You're going to take one cell, divide it into 2, and it's going to, each of the 2 cells, sometimes called daughter cells, each of the 2 cells are going to be just exactly like the parent cell. If each of the 2 daughter cells has 46 chromosomes, that's 23 and 23, like the mother cell had 23 and 23, and there's 2 daughter cells. Where did we get the extra DNA to do this? It was brought in and manufactured during the time before cell division. It will have been brought in during interphase. Interphase is not a part of cell division. It's when the cell is working. It's when the cell is preparing to divide. Interphase is not, like some instructors may have told you, it is not the resting stage. That's the craziest thing in the world, to call interphase resting. Okay, everything is going on. The cell is doing what it's supposed to do. It's either transmitting stimuli, it's phagocytosing other things, bringing things by pinocytosis and active transport, and manufacturing the new chromosomal material. This is a very active stage, interphase. In human beings, the nucleus of normal somatic cells, body cells, contains 23 pairs of chromosomes. You got 1 pair of each chromosome from your mother and one from your father. Okay. Your father contributed to you 23 chromosomes. That's a set. And the mother contributed 23 chromosomes. That's a set. The total number of chromosomes you have, or should have, unless you have a mutation, is 46. And the ploidy number is 23. What the hell is a ploidy number? That's been batted around so many times. If you have a deck of cards, okay, and you've got 4 different suits in a deck of cards, just take one suit. That's a set of chromosomes. How many cards are in one suit, not counting any jokers? How many cards are in one suit? Poker players.
Is it 13?
Yeah. Yeah, I'll sit down at the poker table with any of the rest of the people, though. It's 13. The ploidy number in a deck of cards is 13 because there are 13 types of cards. You have 23 types of chromosomes. You also happen to have duplicates because one set of 23 comes from each parent. The ploidy number, or N, is 23. When we refer to the diploid cell, it's 2N, 2 times 23, 46. Here's a chromosome. Over here and here, there's 2 chromosomes. They're in different colors. I don't care which one you assign to the mother and the father, okay? But all of the genes are located on the chromatid. A single strand of DNA making up one chromosome is called a chromatid. These have a special spot called a centromere, where spindle fibers will attach to pull the chromosomes in one direction or another, separating them later. Here, the centromere is shown in the middle. It could just as easily be at the end or the other end. It does not move around, however. It stays there. The chromosome from your other parent, let's call this chromosome A. And this is chromosome A from your other parent. It'll look exactly the same, have a centromere, and it codes for the same genetic information all the way down. If there's some eye color up here, there's a gene up here, there'll be a gene for eye color over here. They may not have the same quality. In other words, one may code for no color at all, the other may code for blue. And indeed, eye color is coded by several genes in the chromosome makeup of your body. It's not just one locus or loci. Okay. The centromere, I don't have it here, but you're going to see it in your book, I'm sure, has been called the kinetochore. There is a difference, and we will always use the word centromere. So always expect that. I will not use the word kinetochore on purpose. If I do accidentally, I'll correct myself right away. Okay. Okay, so here are 2 chromosomes. How many are in this cell that we don't see around it? Total of 46. Total of 46. There are 22 more pairs of chromosomes. 22 coming from your father, let's say, and the 23rd one that's already in our drawing, 22, 23. 23 and 23, 46. Okay. In getting ready for division, the chromatids duplicate. This chromatid here on the outside is duplicated and another chromatid appears. These will be identical. This chromatid and this one have the same genetic information in the same order. Now here they're called sister chromatids. Even if they come from the male chromosome, they're called sister chromatids. I just refer to them as chromatids. Same thing happens with the other parent's chromosome that matches. The chromatids double. The amount of DNA, or you can say the amount of color that you see on the screen here, is double. It's double. Are the number of chromosomes doubled? No, they are not. Over here we have 1, 2 centromeres. The number of centromeres is equal to the number of chromosomes. There are 2 chromosomes here. Here, 1, 2. There are 2 chromosomes over here as well. This is not 4, not yet. The stages of mitosis, I have a drawing for you. We'll refer back and forth. The first stage of mitosis begins with all of the chromosomes already being doubled and having 2 chromatids apiece. You still have 46 chromosomes, but they are double as far as the amount of DNA is concerned. During the first stage of mitosis, the nucleolus that we saw the last lecture disappears. It was the dark spot. I have the drawings for you, so hang in there. The chromatin condenses. Now the chromatin is largely made up of those chromosomes, but the chromatin during prophase, the first phase of division, condenses like a loose plastic rope, and you twist it tight, 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 tight. It's going to condense. That's to protect everything inside. The nuclear membrane begins to disintegrate, and the centrioles that we saw split into and migrate towards the poles. Okay, we got to now go down and take a look at that. This is interphase here, when that's not a part of division. Prophase is. Here's the centrioles migrating. Okay. They'll go to opposite poles, and they send out what we call spindle fibers. The chromatin that we saw in interphase is really condensing now. You'll notice that there are 2 chromatids for each chromosome. Don't try to match up the colors, not necessary. There are 46 chromosomes, they're all double, they're all condensing. The nucleolus here, the center for transfer RNA production, is largely disappeared. You don't see it in prophase. And the nuclear membrane begins to disappear. Now, some books, some people, some teachers say it disappears, it dissolves. That's, I just take that with a grain of salt. That just tells me they don't know what it does. This nuclear membrane is continuous with the rough endoplasmic reticulum and the smooth endoplasmic reticulum. And it, I think, just disperses out, just spreads out. But in any event, under the microscope, we don't see an intact nuclear membrane anymore at prophase. So we've gotten this far. The main thing is what's happening with the chromosomes. They now each have 2 chromatids, and there are 46 of them. This cell is 2N. It is diploid. 2N because it has 2 of each of the 23 types of chromosomes, for a total of 46. Now we'll go back up. The second phase is metaphase. In metaphase, the cell is also going to be diploid. The spindle apparatus has been set up. Chromosomes are aligned on the equator or equatorial disc of the cell. The chromosomes do not need to pair off at this stage. If you were told otherwise, it's not true. I'm not trying to break myths or anything here. I just want to set the record straight. Chromosomes do not need to pair off, although it can occur. The nuclear membrane has completely disintegrated. Okay, now let's go see what metaphase looks like. Down here, it's this one. The centrioles have migrated opposite each other. The chromosomes, 46 of them, although only 4 are being drawn here, be a mess otherwise. 46 chromosomes, each with 2 chromatids, are on the cell's equator. Now, equator is one word, but a better word would be equatorial disc. Okay. If you have a glass of water half full, so right at the water line, the water touches the edge of the glass in a circle. Okay, that would be an equator. But the water level goes all the way across the inside of the glass. It's a disc. So there's plenty of room for 46 chromosomes, not if they're in a straight line like this, but spread out. Okay. Metaphase. The spindle apparatus, as it's called, stretches from centriole, centriole to centromere. Let's take this one down at the bottom, dark. There's a fiber, spindle fiber, that attaches to the centromere. From this centriole, on the other side, there's another fiber that attaches to the same centromere. There's going to be a tug of war started pretty soon. The spindle fibers are going to retract like fishing lines, and they are going to pull the centromere apart and take with them one chromatid this way, one chromatid that way. Now, when I say they're going to pull the chromatid this way and a chromatid that way, each of the 2 pieces is going to have at least half or approximately half of the centromere. Whether you're talking about a centromere or half of a centromere because it's been split, you're still talking about a centromere. So going in this direction will be a chromatid with a centromere. It's a chromosome. It's still a chromosome. And a chromosome goes this way. You can talk about them as chromatids with partial centromeres. But if there's any part of a centromere present, it's a chromosome. So what's going to end up happening here? These are all lined up. They're not paired up, but they're all lined up. When the lines are drawn in, all of the centromeres are going to split if it's done properly. And 46 chromosomes go one side, and 46 chromosomes will go the other way. That's going to show in this next diagram, but we need to back up and say that the next phase is anaphase. This phase is a little more, not complicated, a little different. The cell is said to be temporarily 2n plus 2n. That's because all 46 chromosomes split into two sets of 46 chromosomes. And one goes, let's say, to the right, one goes to the left. We'll have 92 chromosomes temporarily in two groups, 2n plus 2n. The centromeres split. If you could underline this somehow for yourself, I should underline it, make it bold. The centromeres split. That doubles the number of chromosomes. Each chromosome that results in the splitting of the centromere takes with it half of the chromatids. And then when the centromere splits, it's considered that the result is two centromeres or pieces of centromeres. Chromosomes migrate then towards the centrioles, half of the chromosomes to each one. Okay, here we're set up. We're at the line of scrimmage. Okay, and now their signal, whatever it is, don't know, is given. And the centromeres split, and the resulting part centromere and chromatid, which is still a chromosome, is drawn to one centriole or the other. This whole group goes this way. This whole group goes this way. This whole group consists of 46 chromosomes. This whole group consists of the same number, 46. This can't last long, this 2n plus 2n situation. And we've got to have a division of the cells. In the animal kingdom, us, the cells divide by furrowing. It's like tightening a belt. Or you can sometimes, if you've ever blown bubbles, get bubbles to attach to each other, yes, but you can also make two bubbles out of one by furrowing. You end up with one centriole on each side. You end up with the 46 chromosomes here and the other 46 chromosomes here. There's no crossing over, there's no recombination, there's no genetic instability. This cell that's in the formative stage is exactly genetically like this cell. This is telophase. Telophase, this is the cell phase when we have cytokinesis, which is cell splitting. And then it finishes off. The nuclear membrane reforms. The chromosomes loosen up and become chromatin again. The nucleolus returns. Nuclear membrane returns. So we have this cell and this cell. We have two cells, each looking like the first cell, with one exception. This one has two centrioles. Down here, the two new cells each have only one centriole, but the centriole is capable of duplicating itself, and it will prior to the next division. Same on this side. So these are exactly the same. Mitochondria, important organelles inside of the cell, also are capable of reproducing. If all the mitochondria ended up in one side just by happen chance, or if that's the word, the other side would die. And so you have squandered one cell division. But that's never likely to happen. The mitochondria will split. And what happens to the ER and the smooth endoplasmic reticulum? Hell, it fills everything. And so when you have furrowing occur, you'll have splitting of the endoplasmic reticulum as well. Can't miss. Okay, I'm going to give you a 10-minute break, just a short one if I can. Looks like it's 3 o'clock, so let me stop the sharing. Now I can see it. Yeah, it's 3 o'clock, so 3:10. And I'll see you in a few minutes.
Thank you.
Okay. Caleb, are you here? Can you guys hear me?
Yes.
Okay. Caleb, are you here? People who put up black boards.
Yes. Yes. I'm
Okay. Thank you.
Yeah.
We'll be off of here pretty quickly, but if you can put your video on for a second. Okay. Thank you. Thank you. I've seen Kareem today and Sandra I've seen today. I haven't seen Katie. I don't know if I've seen Emily Shard. Okay, anybody with a question before we get back to it? Okay, what we're going to look at now then is meiosis. I'll be so happy when we're finished with this. I'm sure you will too. Stages of meiosis. Meiosis is known as reduction division because it is the type of division in which the number of chromosomes is cut in half. This type of division, meiosis, is only found in the ovary and in the testes, where gametes are formed. The gametes having half as many chromosomes total. They will have 23 up in each gamete instead of 46. Okay, in the stages of meiosis, which, as I said, occur only in the ovary or the testes, prophase 1 is similar to meiosis 1 with an important difference. In prophase, the chromosomal material is loose. It's chromatin. The chromatids of paired chromosomes can overlap numerous times. Remember how we had 2 chromosomes that were identical, one from each parent? Okay. Well, we had them each form 2 chromatids. We still had 2 chromosomes. Okay. Back up, back up. This situation happens in meiosis as well. Your 46 chromosomes become 46 doubled chromosomes, double meaning 2 chromatids. And in meiosis, okay, the male and the female chromosome that are homologous. That means, for instance, the ace of spades, the ace of hearts, the king of spades, the king of hearts. Okay. Homologous chromosomes are like also on TV. Chromosome A from the other parent, and then chromosome B matches up with chromosome B. They find each other. They find each other, and these arms of the chromatids can swing across each other and then pull back. And in doing so, exchange genetic material. The top of this one can now become the top of this one. What happens to this one? It goes over here. It's just sort of like a sleight of hand. A lot of times the book will show it happening only once, but then it can happen again and cross over farther or farther still. It's like shuffling a deck of cards. The more you shuffle it, the more random everything is going to occur. And so you're going to get randomization. This chromatid may share or shuffle with this one. This one may shuffle with this one. How? How's it going to reach way the hell over there? These are not stationary on a piece of paper. They float. This one can turn, float over, turn upside down, and touch this one and this one. So all of the chromatids can exchange genetic information. Okay, back to prophase 1. Okay, why do they call it 1? Because there are 2 divisions, 1 and 2. In prophase 1, the chromatids of paired chromosomes may overlap numerous times, with the subsequent exchange of genetic material between them. Recombination of genetic material occurs. Okay, that's important. Recombination, shuffling, occurs in the prophase 1. In metaphase 1, the chromosomes are aligned on the equator, just like they were in mitosis. Chromosomes are paired, each near its partner. Interphase cell, just like any other. Prophase 1, the centrioles move. And in prophase 1, the DNA is doubled. Each chromosome develops a second chromatid. That's where the doubling occurs. And the crossing over, the recombination occurs here as well. The nuclear membrane breaks down. The nucleolus is disappearing. They still show it a little bit right there if you can spot that. And that's the end of prophase 1. Okay. Then at metaphase, the chromosomes line up. They find their partner. In other words, chromosome A from your parent, father, finds chromosome A from your mother. And they'll be close together. You'll have 23 pairs. 23 pairs. And then, as we'd expect in anaphase, the chromosomes split. Okay? The chromosomes move to different sides. the chromosomes move, and I said the chromosomes split, but I don't want to confuse you here. The centromeres do not split. The centromeres do not split in anaphase 1. Instead, a recombined, shuffled, genetically different chromosome goes one side, and the other chromosome goes the other side. Then, in the next instance, the nuclear membrane reforms around them in telophase 1. Furrowing occurs. The centrioles are found on either side. The reason they're showing these chromosomes in different colors, here, light blue, dark blue, is to show you that they have been recombined. They've only got one little bit recombined, but there's lots and lots of recombination that can go ahead. If you take your hands like you were clapping them, okay, but instead of just clapping, overlap your wrists, overlap, and then pull back. It's possible that if you do that enough times in your chromosome, your right hand becomes your left and your left becomes your right. You have exchanged. Now, cross your hands again. But this time, halfway up your radius and ulna, cross them there, cross them farther, pull apart, and you'll recombine a different portion. You are really shuffling the deck. At the end of meiosis 1, okay, since you did not break the centromeres, then half of the chromosomes went one way and half the other way. That means you had 23 chromosomes going to the left here and 23 chromosomes going to the right. And each of the new cells has only 23 chromosomes. That's half of the 46. These are haploid cells, also known as 1N, because they have one of the ploidy set of chromosomes. But then they will revert back to what looks like a regular cell, except recombination has occurred and there has been a reduction in the number of chromosomes. Let's go up and read it for a second. At metaphase, the chromosomes are aligned on the equator of the cell. Chromosomes are paired, each near its partner. Then in anaphase, the centromeres do not split. They did in mitosis. It will not do it in meiosis. In meiosis, the centromeres, and I did use the word kinetochore here, but I'm not going to put it on a test. The centromeres do not split. Chromosome pairs separate, move to opposite poles. In the telophase that follows, it's a short phase. It's similar to mitosis. Reduction and recombination both occur in meiosis. I'm going to ask that. That ends meiosis 1. Now these are only 2 cells, and you're going to get the formation of 4 cells. How? Here are the 2 cells. We don't need to look back at the text above. In the prophase 2, okay, you get the reestablishment of the chromosomes again. It was back to chromatin. Now it condenses again. But these are genetically modified chromosomes, and there are only 23 in each cell. Centrioles develop in both cells. They form like this, and like this. We only have half as many chromosomes over here, 23, 23 over here. And if you'll notice how one part of the chromatid, one chromatid is different than the other chromatid. They've only shown it different in one respect, one area, but it will be different in hundreds and hundreds, if not thousands of areas. And now at anaphase, the centromeres do split. A chromatid with its centromere portion, properly called a chromosome, will go this way. A chromosome goes that way, that way, that way. Seems like we're doubling up the number of chromosomes again. No, that won't happen because furrowing happens immediately after and keeps the number in half. The result after cytokinesis, which is the splitting of the cell, is a haploid number of chromosomes, 4 daughter cells, each having one set of chromosomes, a complete set. Reduction has occurred. We don't have 2N, we have 1N. Every one of these cells that I'm pointing at has the potential to form a gamete. If they're all equal as they are here, it has to be in a male. Females don't have equal divisions, you'll see in just a second. And these will form spermatozoa. Every spermatozoan is 1N or haploid, and every one of them is different from every other one. The odds of them having the same chromosomal makeup are unbelievably high. Not going to happen. It's not going to happen. They're not going to have the same genetic makeup. They're going to have the same gene combinations, and they'll have a lot of characteristics in common. So we possibly expect siblings, for instance, to look fairly much alike. But they're not going, these are not identical. This is not going to lead to any identical twins. Okay. If there's a dominant character, such as a birthmark, that can run through the family, yes. But all of these characteristics mixed up as much as they are, it's like taking 4 decks of cards and shuffling them and expecting that as you take the top card off of each of the 4 decks, it'll be exactly the same for all decks, not a chance. Remember that the reduction and the recombination occur in prophase 1 that's of meiosis 1. Meiosis 2 is just a further sectioning to the 4 cells. In females, the divisions are not even. They are uneven. You got to remember, when an egg and a spermatozoan or sperm cell get together, that sperm cell only has genetic information in its head, and it's tiny. It's got a little tail. The egg of the female is like, I see the sperm cell coming in as a rocket, an exploring rocket coming into the sun. The egg is huge. The egg has genetic material also, though, and the genetic material of the spermatozoan is complete. If the two fertilize, then you'll have the formation of an embryo. In females, the divisions are uneven. The first division leads to one large cell and one small cell, not, as I'm pointing here, to two even cells. One large cell and one even, much smaller cell. That smaller cell is called a polar body. It will not survive. It may be able to do the second division and form two more polar bodies. And the large first cell can form one more polar body. So you can get 4 cells in the female, but one only is viable as the egg. And the other 3 are so small, they can't survive. They have next to no organelles inside of them. The polar bodies do not function. In males, backing up here, the sequence of the 2 meiotic divisions lead to 4 morphologically identical spermatids. In other words, they look alike, but they are not alike genetically. They have to differentiate, change their shape to become spermatozoa with a tail and a neck. All will be genetically different. A couple of terms. In meiosis, we have gametogenesis, the formation of gametes or sex cells. All gametes are 1N and are genetically dissimilar. In the male, the process of gametogenesis is called spermatogenesis. This is the formation of sperm cells, and it continues in men from puberty until death. In females, the process of meiosis is called oogenesis. This is the formation of ova, and this is complete at or just after birth in the human female. So in a female baby, the ova are already present. There won't be any more. There shouldn't be any more anyway. When people decide that they need to stimulate the ovulation to gather ova, for in vitro fertilization, because they can't seem to have kids any other way. When you do that, you stimulate a lot of ova, and a lot of them don't survive the process. There's no replacements for those. And once you've taken all of them, there are no replacements for them. So hopefully it works. Okay. Okay. I'm back at week 3, and this is the lab, but we have plenty of time on Thursday as well for the lab because I'm not going to lecture on Thursday. There's nothing on Thursday that is going to be on the lecture exam, but the bones obviously are. So I don't want to rush through them. I'll go through possibly about half of them today and half the very first part of Thursday, and still leave plenty of time for review. Here's our figure locations. So with your book, you can see the bones that we're going to look at. Yeah, that would be a lot for this afternoon. Anybody that wishes certainly can go ahead. I think I like my method. And okay, these are the bones of the lower appendicular skeleton. Remember how many bones are in the appendicular skeleton?
$126.
Yep, that's it. I couldn't get to sleep the other night. I tried counting the number of axial bones. I know there's 80. And if I just look at the spine, I already know that's 50. So I only have 30 to come up with. And I always too short. And it's never the same two. Keep trying, and then finally I fall asleep. This is sometimes called the coxal bone. That's not a good name for it. It's okay for us to talk about it as coxal bone. Here's the word here. But there's no such thing as the coxal bone. This is a fusion of 3 bones. The 3 bones of the ilium. Okay. If you put your hands on your hips, okay, you know, in that pose, if you're disgusted or what's going on, put your hands on your hips. You're touching this portion here, the iliac crest. The iliac crest, that goes behind your back as well. Okay, the iliac crest. If I put my cursor right here at the iliac crest, and now I want to go forward. How do I know that's forward? Here's where the femur head is going to go. And the femur is going to dangle off on this side. Okay. Back here, a broad piece of bone that we sit on. This must be the posterior. The anterior is here. So we follow the iliac crest to this point. This is the anterior superior iliac spine. After a couple of times, you'll get it. At first, it seems like a mouthful, but very few people ever miss this. Drop down about 1 inch to 1.5 inches. And this is the anterior inferior iliac spine. You're looking from the back. You're looking from the posterior in this view. We'll get at the other view in just a second. I want to go back up, across. Well, actually, I want to dwell on these two for a moment. The anterior and superior and anterior inferior iliac spine. These are what you hitch your belt on. That's what we commonly say. I don't know, everybody doesn't have a belt anyway. Some people use elastic. But you've all seen people, thin people, at the beach in bikinis or Speedos, and they got 2 bumps on the right, 2 bumps on their left side. Okay, those bony projections are the anterior spines. Come over to the other side. You can't feel this on yourself. You can see it on the model. This is the posterior superior iliac spine. Go under it, drop down 1.5 inches to the posterior inferior iliac spine. With your finger still on that, go into this deep crevice. This is the greater sciatic notch. Keep on the edge of the crevice and you'll come to a point. This is the ischial spine. We've left the ilium and we're in the ischium. This is the ischial spine. You'll see in a moment just where these separate, all come together right here in this cavity. The ischial spine is important because if it sticks in too far in a female, it'll be difficult giving birth to a child. Find that ischial spine on your screen, come down under it, and there's a small indentation, much smaller than the greater sciatic notch. This is the lesser sciatic notch. Okay, this receptacle for the head of the femur is just about the right size to set a golf ball in. This is the acetabulum. This large hole through which muscles and nerves and blood flow is the obturator foramen. At the posterior, I mentioned it a moment ago, this is what you sit on. If you've ever had saddle sores from a horse, this is the bone and on the other coxal bone as well. This is the areas where you're bouncing. This is the ischial tuberosity. If you've ever sat in the bleachers and the seats were hard, it's the ischial tuberosity that suffers. And then down here, right in the front, is the pubis bone, also known as pubic bone. Here we have everything put together. So we have a couple things. Here's our iliac crest, not labeled, but in the anterior view, we have this rather smooth indentation, large. This is the iliac fossa, and it's in both diagrams. The pelvic rim is this circular area, sometimes oval. Okay. In the female, it tends to be oval. It's the pelvic rim. In the male, it tends to be more circular, pelvic rim. The ischial spine that we looked at's here or here. We've got to put the sacrum in. Okay. And the sacroiliac joint, not labeled here, is right there. Sacrum, iliac, ilium. If we look at the underside below the pubic bone, this is the pubic bone. In the female, this angle is usually 100 degrees or more. And in the male, it's definitely smaller. It says 90 degrees or less, but every skeleton I've looked at, it's about 30 to 40 degrees. Very sharp, very sharp. Whereas the female has an arch that is wide. Here's that femur, the head, the neck makes things easy. Let's look at this side, head, neck, and then we have the greater trochanter, which is lateral. Remember in the humerus, we had the greater tubercle, and it was lateral. Here's the greater trochanter. Here's the lesser trochanter. These are the primary spots for attachment of leg muscle. Looking at the posterior side, greater trochanter, lesser trochanter, but the moon that we see there is the intertrochanteric crest. There's an indentation in the head where my cursor is, and it's pointed to over here. This is the fovea capitis. It's a true marking. In other words, it's something we can point to, but we don't really have a use for. Everything that has a structure does not have a function. This indentation, fovea capitis, means literally hole in the head. Let's come down the posterior side, and there's a very sharp ridge. This here, down to here, is the linea aspera. It will be the point of attachment of our hamstring muscles when we do the muscles. Nothing to worry about yet. Linea aspera. Did we have something like that on the humerus? Yes, we had the deltoid tuberosity. So a lot of similarities. Come down the posterior side again. We had the olecranon fossa on the humerus, but here we have the intercondylar fossa. Intercondylar fossa. There's a medial condyle and a lateral condyle on either side. How do we know which is medial and lateral? We go up here. The attachment of the leg to the pelvic region. This has to be medial. This is lateral. Medial condyle, lateral condyle. And now go back to the anterior view, the anatomical view. Come back here. There's a smooth area. just one smooth area. This is the patellar surface. Fortunately for us, we don't have capitulum, radius, radial fossa, coronoid fossa, things that we have in the humerus. In the anatomical view of the femur, we just have the patellar surface. And the patella, of course, kneecap right in front of it. Okay. Head, neck, intertrochanteric crest, it was called earlier. Here it's being called line. Don't worry about it. It can be either, but the word intertrochanteric will always be there. Lesser trochanter, lesser trochanter, greater trochanter. We're not doing the gluteal tuberosity. How do you know? Because if you have that sheet that I showed you just a while ago, it's not on there. Come down, intercondylar fossa, medial condyle because it's on the head side, lateral condyle because it's on the greater trochanter side. And on this side is the patellar surface. And again, the lateral condyle. Over here, the two bones of the lower leg are put together. We need to see them again. This isn't good enough, but it's good. Here's our tibia. You can't call it shin bone in the class or on the test, but that's what it is for our sake. This one is a right tibia, and next to it, a fibula. The fibula is always lateral. You got to commit that one to memory. The fibula is always lateral. If you get hit from the side, in sports especially, called clipping, and you can injure or break the tibia, the fibula, quite easily. At the top of the tibia, there is a double peak. It looks a little bit like saddleback. Saddleback has two peaks, Santiago and Majeski. Okay. Those two peaks are the intercondylar eminence. Eminence because it's pronounced. Intercondylar because it's between these two condyles. This is the lateral condyle. How do you know? Because the fibula is always on the lateral side. We just said that. We don't even have to look further down. This is the medial condyle. We can look further down though. Take the tibia if it's by itself. At the distal end, grab a hold of that small piece. You can barely hold the bone up doing that, but you can. This is the medial malleolus. Left to our own devices, we would mispronounce that word all of the time. Okay, this is the medial malleolus. At the end of the fibula is the lateral malleolus. And these two together make an arch that helps to hold the foot. Sorry. Here's the tibia. Right around here is the anterior border. The anterior border I know you've done it. You've hit your shin, or you got kicked in the shins. I usually hit it on the side of the bed or something. That point in the front of the tibia is your anterior border. They're pointing out the interosseous membrane. This won't appear on your practicum, but it will appear on your lecture test, because this is a slightly movable fibrous joint, a syndesmosis. It's the one we use as our example between the fibula and tibula. Don't bother to learn it as interosseous membrane. What you want is syndesmosis. Don't worry about proximal tibiofibular joint, which is right there. The upper portion of the fibula, though, is the head. And the lower portion is the lateral malleolus. I'm going to skip over the knee because I'm going to do that one in detail. We'll finish off the day with the foot. And if you think I say the hardest for last, it's not true. Foot's darn easy. Okay, easier than the hand, I think. Okay, the foot has obviously toes. Okay, and each toe has 1, 2, 3 phalanges, with the exception of the big toe. The big toe is your anatomical position is always medial. It has 2 phalanges. It has the proximal or phalanx and the distal phalanx. All the rest of the toes have the proximal phalanx, middle phalanx, and distal phalanx, just like the fingers. So we wipe out 14 bones right there, 2 feet, 28 bones. If somebody steps on your toes, this is what they're stepping on. If somebody steps on your foot, it means that they got up here, okay, with the metacarpals. I'm sorry, the metatarsals. Okay, metatarsals. This large metatarsal here, the largest one, goes to digit 1, the big toe. This is metatarsal 1. And you guessed it, 2, 3, 4, 5 for the little toe. Metatarsal 1 coming back towards the heel articulates with 1 bone. If you remember back to the hand, metacarpal 1 articulated with the trapezium, just 1 bone. Well, here, same situation, but this 1 bone here is called cuneiform 1. It can also be called the medial cuneiform. Do yourself the biggest favor and learn it as cuneiform 1. You'll see why in just a second. The second metatarsal articulates with cuneiform 2. The third one with cuneiform 3. There's cuneiform 1, 2, 3, and then the 1 here articulates with the last 2 digits, just like in the hand. In the hand, this would have been the equivalent of the hammy, but this is the cuboid bone. Cuneiform 1, 2, 3, and cuboid. If you want to learn it the other way, which is correct, you could call this the medial cuneiform because it's towards the medial side of the foot. This would be the intermediate cuneiform. And this would be the lateral cuneiform. But if this is the lateral cuneiform, what's the hell is this? It gets confusing. This is still the cuboid bone. I think it's much easier for you. 1, 2, 3 cuneiforms. Come back to the last one, and that's the cuboid bone. And come straight back. This articulates with the largest of the bones in the foot, the heel bone. This bone is the calcaneus. It's also shown down here, calcaneus. Sitting on top of the calcaneus is the talus. Here's the talus. Nice smooth surface here to articulate with the tibia and fibula. One bone left. Go down the front slope of the talus, and we have this boat-shaped, upside down but boat-shaped bone. This is the navicular bone. We had a navicular bone in the hand. In either place, hand or foot, it can also be called the scaphoid bone. But navicular, I think, is really caught on now. As I said, I'm saving the knee. And oh, sure. Okay, anybody with a question? Okay, I'll get that email out that I promised you about when things are due, just so that there's no confusion. And if you want to hang around and ask a question, please do. And if you don't, well, I'll see you on Thursday.
Thank you.
Thank you.
Bye-bye.
Thank
Bye.
you.
You.
Thank you.
Bye-bye.
Thank you, Charlie.
Bye.
I have a question.
Sure, Andrew.
Are we gonna be doing the cells on the lab practical?
No,
Okay, cool.
nope, that's lecture material.
That's what I thought. Cool.
You've already taken the third practice test.
Yeah.
How many tries? I didn't look
I think
at that.
3.
3.
I thought I got more right than I did on the first one, but I didn't.
Okay,
Took me 3 times. Even like looking at the answer took me 3 times.
that's okay. That's
Yeah.
why you get extra, tries.
Yeah.
Okay,
All right, thanks, Charlie.
bye-bye.
See ya.